Hello, welcome to this video on transcription, where we'll first talk about the location of transcription. So transcription is going to occur in the nucleus of the cell. So if we're talking about eukaryotic cells. So we start with DNA, and DNA is used to produce our pre-mRNA through transcription. Next, we have this diagram to show you a visual of the central dogma. So we see that pre-mRNA is going to go through RNA processing in order to produce our final mRNA transcript, which is going to exit the nucleus through a nuclear pore into the cytoplasm. Then the mRNA goes to a ribosome and interacts with it to produce a polypeptide. And this process of producing the polypeptide by utilizing the mRNA is called translation. And remember, a polypeptide is just an amino acid chain, and the tRNAs are going to transfer the amino acids to the ribosome. So, good review here of the central dogma, which we'll discuss in more detail in later videos. So the point is that transcription occurs in the nucleus, right? And here is a rough overview of the 3 steps of transcription. The first step is called initiation. The second step is called elongation. And the third step is called termination. So in the initiation phase of transcription, the first thing that's going to occur is the transcription factors here, kind of colored, are going to bind to a region called the promoter. So in green right here. So the promoter region is on the DNA, which we have right here. And this allows or attracts, in another word, an enzyme called RNA polymerase to the promoter region so that it can bind to it. Okay. So altogether, the RNA polymerase right here and the transcription factors that are bound to the promoter region, this whole unit is called a transcription initiation complex. And this transcription initiation complex is important, or it's the main component that's going to drive transcription, right? And we can see transcription initiating right here by the formation of our mRNA transcript, right? Okay, so as you will also notice, we have 2 strands of DNA, one DNA strand going from the 5' to 3' direction, and the bottom DNA strand going in the 3' to 5' direction. So the 3' to 5' DNA strand is called the template strand. And this template strand is going to be the strand of DNA that's going to be used by the RNA polymerase to produce the growing mRNA strand right here. So it's only the template strand. The strand above is called the non-template strand of DNA. Okay, so let's move on to step number 2, which is called elongation. And it's as simple as that, as the name of the step. So elongation is basically when the mRNA transcript is going to keep on elongating. It's going to get longer, right? Other direction, right? And I put a note right here. The mRNA is still going to grow from the 5' to 3' direction, utilizing that template strand that's in the 3' to 5' direction. And who's adding the nucleotides for the RNA? It's going to be RNA polymerase that's going to be doing that. Okay. So is this going to stop? Is this transcription ever going to stop? And the answer is yes, when we reach the terminator sequence right here of DNA. So when the RNA polymerase reaches the terminator, right? This is going to result in the mRNA strand detaching from the DNA, and also the RNA polymerase is going to detach from the DNA, and we have our complete, in this case, pre-mRNA, right, transcript. And then this pre-mRNA transcript can now go through mRNA processing to produce our final mRNA transcript. Okay. So hope you enjoyed and hope you found it helpful. Please like and subscribe and follow for more content. Thank you.